Good afternoon, everybody, and welcome to another instalment of the COE seminar series. I would like to acknowledge that this meeting is being hosted on the traditional lands of the Bindale people and pay my respect to elders both past and present. And remind everyone that our expectation is that interactions during seminars are constructive and that all attendees behave with respect and consideration for others. Thank you very much. And just a couple of Zoom housekeeping things before we get started. I'm sure you're all very familiar, but if you could just make sure that your microphones are on mute for the duration of the seminar, that would be terrific. And there'll be an opportunity at the end to ask questions, and you can take yourself off mute and turn your video on at this time and ask questions directly, or you can put them into the chat box and I'll help read them out and ask for Bert's responses. So it gives me great pleasure to introduce Robert Streit as our speaker for today. I've known Bert for some years now, but my first real introduction to Bert's work was at a conference, I think it was ASFB, where he rather dazzled the attendees actually with the talk about how fish use space, so what he's going to be talking a bit about today. But the really cool thing about it was that he managed to communicate some quite complex work in a way that was both really digestible but also visually pretty impressive. And from talking to Bert recently, you know, he was saying that this is one thing that really interests him. Like, yes, he enjoys working on fish and making maps, but he's also really interested in, you know, breaking down complex ecological processes and communicating them in really engaging and digestible ways. So I'm excited to see Bert's talk today, and I'm also really excited that Bert's recently started a postdoc at the Reef Function Hub at JCU and COE, and he's been based in Brisbane for a little while, but he's about to move back to Townsville. He's here in Townsville now, but he's about to move back in a more permanent capacity. So I hope you'll join me in welcoming Bert for this seminar, but also back to Townsville when you see him about. I'll hand over to you now, Bert, to take it away.
Cool. Well, thanks very much, Eva. Very good way to increase the expectations. But yeah, good summary. Like, as Eva said, I really enjoy trying to make complex things easy because that's the only way I can understand them. And that's what I'm trying to do today with this talk. So the title is Space Use by Reef Fishes. That's my PhD thesis title as well. But for this talk, I've added this subtitle that is more than the science of where. And now I'm trying to make it understandable what I mean by that in the next sort of 40 minutes. So as we all know, reef fishes are really important on reefs for all sorts of different things. And that's what I work on. But while that's true, that's not really the talk I want to give today. So I'm not really today wanting to talk about the results of my work, really, but rather also because of the daytime, it's after lunch. So a lot of us are doing a lot of digesting already. So I thought I'd keep it quite light. And I'll talk about fishes in space. And obviously, while I don't talk about actual fishes in actual space, what I want to do today is, as I said, not talk so much about the science and the results necessarily of my work, but sort of about the stuff that sits behind it or above it. And part of that is ecological theoretical concepts. And I'm sure you're super excited to hear that that's what we're going to talk about, as much as I am, really. But apart from that, I also want to talk about methods, because methods are often seen as tools for answers. So you have a certain question and you employ a certain method to answer that question. But what I sort of stumbled into during my PhD was this other thing, that methods can also be really good tools to ask questions. Because if you do something in a new way, you might get a new perspective on your question, and you might be able to actually come up with completely new areas of questions. And that's what happened to me in my PhD accidentally, and that's sort of what I want to share with you today. So when we think about space use and spatial ecology, the first question is, what is that? So what is space use? And to start off with, let's get really awkwardly personal. So here you can see Townsville from a satellite image and a pin in around South Townsville. And that pin is actually me. So this is my movement tracked through my phone, through Google Maps for about a week. And it was in the last year of my PhD, and you can tell it's the last year of my PhD because I'm basically just moving between where I used to live and uni. So there wasn't a lot of exciting stuff going on. But what you can do, you can track that movement and you can see, sort of quantify that over time. And through that, you can then draw a boundary around it. And if you're ballsy, you call that a home range, or you just call that a occupational, a space of occupation. And so that's the basic question. If you think about spacies, the first thing you can ask is, where are they? Where are the fishes, or where are the PhD students? And that's sort of the first question. And with fishes, we can't give them mobile phones and Google Maps, but what we can do is use this technology, and that's acoustic telemetry. So with that, you put these microphones essentially underwater in certain positions you're interested in, and then you put little transmitters, you implant them into fishes, and then you can track where these fishes move around on the reef. And that's a really useful technology to study space use because it's long-term. Those batteries last for up to years. It's inherently space-based because you put these receivers into locations of interest, and you track fish movement by doing that. And there's a lot of studies over the years that have used this on reef fish. And I'm just going to introduce this one. This was done by Justin Welsh in our lab almost 10 years ago now. And he worked on this big parrotfish. So that's a big Chlorurus microrhinus, and they grow up to 50, 60 centimeters. In the bottom image there, you can see it for scale, and you can see the little transmitter that gets surgically implanted. So these fishes are quite big, and Justin wanted to know how far they move and where they live. So his study was on Orpheus Island. This is Pioneer Bay. So the JCU research station is in the bottom right corner there. And that's where, in that bay, he tracked those fishes for 7 months. And these are the results. So these sort of fried egg shapes are the home ranges of these parrotfish. Each egg is an individual home range. And the first thing to note is that these fishes really have homes. They don't just randomly move around the reef and sleep here one night and there the other. They have distinct places where they live. And also these home ranges are quite small. So the extent was around 8,000 square meters, but the core area, the gray bits, were only around 1,700 square meters. So really not a big space for a fish of that size. So as I said, there's many studies doing this, but what a general theme behind these is that fishes have small and stable homes. And that is really the where. So that's the where of space use. You can figure out where these fishes are. And that's a really important starting point, because any other questions you might have to grow from that location. But really, when I started my PhD, that wasn't quite enough for my brain. And that was because I started in 2016 when we had the big coral bleaching event on Lizard Island. And these are photos that we actually use in a data set, and I took those in my first PhD year. And what really struck me was the speed of the change of reef. So home ranges are good to know what's happening in the status quo, but seeing how quickly reefs can change in their composition, I wanted to know a bit more about spatial ecology. So obviously most of the audience here knows that reefs can change fast and have changed recently from coral cover, from high branching coral to less coral and to more turf algae. But what I also want to point out is that it's not only a quick way down, but we can also see regrowth, and we can see actually quite nice reefs. So this was January this year on Lizard Island. But this was also January this year, just a few hundred meters further. So what I want to point out by this is that we can have this quick change and this patchiness in reef condition, both on the way down and again on the way up. And we have to understand what this means for fishes, because we know certain fishes have habitat dependence on corals, like these damselfish. These are coral obligate damselfish that live in coral. And as far as we know, they need that coral to survive for shelter and to get their food. And without the corals, we assume that these fishes disappear. So if this coral is being lost more and more, we have to understand what happens to the fish. And then also in terms of ecosystem processes, because we know that parrotfishes, for example, feed on algae and can help improve reef stability by removing or apprehending algal overgrowth. But if we have this increase in areas that are covered in algae, we have to understand whether these fishes can keep up with this. And that question was sort of the core of my PhD. Can fishes keep up? And to put this into a bit of a more colorful and conceptual way, let's have a look here. So the question is still, can fishes keep up? And we have this nice reef, and then we have a happy fish living on that reef. But then imagine a disturbance comes along, in this case, illustrated by macroalgae, but it could be bleaching or crown of thorns or cyclones. And that hits in certain spots. And then around that disturbance, we get a degradation of reef condition. And then over time, that can degrade in other areas and so on. We can get more disturbances. And if we really look over a long time, we can end up with this really patchy mosaic of reef condition, just given the different disturbances that act on different scales and also different recovery trajectories. So we have to understand what happens to the fish that live there. Would these fish just disappear and we end up with a really sad fish community because the conditions are so bad? Or alternatively, we have to understand whether these fishes might be able to move and really readjust to this patchiness. So to put this in a bit more scientific terms, what we really should look at is heterogeneity or patchiness of reef condition. And on the other side, connectivity, to understand whether fishes can link and bridge these differences in reef condition. So for me personally, what I'm interested in is assessing space use of fishes while explicitly acknowledging the patchy and dynamic shifts in habitat condition. So that was sort of the main core of what my brain has done in my PhD. So this is the background, and we talked about the where already. We talked about home ranges, where fishes are. But if we're interested in this patchiness and connectivity, what we should also look at is where fishes can go. And again, here's an example from, oh, hang on, no. So why are these movements interesting? Why would that be useful? Well, I've introduced that with the patchiness already, but in terms of ecological theory, this movement is important because these moving fishes could be mobile link species, and they could provide this ecological memory to disturbed sites. So they could transfer processes or resources and in a way provide this functional spillover into degraded locations. And on coral reefs, that could mean, for example, that we get an additional level of response diversity, because if we have a localized disturbance that also takes out the fishes, if we have fishes that can move further, they can come in, fishes from somewhere else can come in and replenish the local function. And a study assessing this mobile linking is again one by Justin Welsh, again done with telemetry. And he looked at Kyphosis vigiensis, which is an important macroalgae feeder on the Great Barrier Reef. And the study was again on Orpheus. So this is the entirety of Orpheus Island. And this here, this is the study I showed you earlier. So the big parrotfishes, they spend most of their lives within those little white blobs. But these Kyphosids, these fish, Justin found that within a day, one of them moved this far. So these fishes really have this potential to move really far distances. Up to 11 kilometers was the maximum in this study. So that means they have the potential to be this mobile link. And to show you this is really exceptionally far, here we have a graph of home ranges on the Y and body size on the X-axis, and the black dots are herbivores. And this is a herbivore, Kyphosis, but he doesn't sit down with the other herbivores. He sits all the way up here together with pelagic predators like barracudas, for example. So this herbivore is really unique and has the potential to be this important mobile link between habitats. So this is mobility through space. So we looked at how far they can move. But if we think about my question from earlier, can fishes keep up? A big part of that was that shiftingness and the patchiness that comes up over time. So we should also really look at mobility through time. And what I mean by that is basically I'm interested in the temporal mobility of communities. So we have to understand, if a community of fish sits in this reef and that reef gets disturbed, does it have the potential to move somewhere else, essentially? And to understand that, I was interested in when homes become established in fishes, because we know fishes have their homes, but we need to understand when in their lives do they choose where they live. And so we could imagine a little juvenile fish that has a nice home, but then there's maybe another home that's just as suitable. And what we need to understand is how does it pick where it lives? Because that'll give us ideas for the long-term trajectory of locations on the reef. And this is what I wanted to get to, but this is really hard to measure because you would have to track individuals throughout their entire lives, essentially. But what you can do, you can get to this via homing behavior. You can use that as a proxy for site attachment. And that works because if you displace a fish and then see whether it comes home, the homing journey is usually quite scary or dangerous. And if they actually commit to it and come home again, that means they must really like the first place. So that's what I did early in my PhD. That was my first chapter. And I looked at juvenile reef fishes. So you can see here some examples. These are a wrasse and a damselfish, and they're really small. So those are my fingers. So you can imagine it's only a few centimeters long, those fish. And we caught them and tagged them with those colorful tags you can see on the tail there. So we could identify individuals again. And then after tagging them, we displaced them. So we took them and put them somewhere else, and up to distances of around 3,000 body lengths. So quite far, really. Again, if you imagine a human toddler, possibly around 1 meter tall, 3,000 body length is 3 kilometers. And whether they come home again is a study I'll let you try and do. But it's just saying it is quite far. And after displacing them, we then went back diving every day and checked where these fishes were. So we basically recorded whether they had come home again or whether they stayed at the sites that we put them to, which were also suitable as far as we could tell. And these are results. So on top there, you see that there are 7 different species I did this on. So it was different genuses, different functional groups. And on the x-axis, you can see body size for each species. And on the y-axis, you'll have probability of homing. And what we found is that across species, we had a really consistent pattern. So across all these different species and different diets and whatever, different ecologies of these fish, we found this consistent pattern that fishes, as they became bigger, they were more likely to come home again. And while you could argue here that small fish are just too small and they don't survive anyway on reefs because they have high risk of predation and they might just die anyway. But so we looked at that as well, and we found in this here, we found a trend that smaller fishes were more likely to die or disappear from our study, but it wasn't significant and the trend wasn't as strong as the homing trend. But what we did find was the third category that we looked at was staying. So that's staying at the release site. And that had this really nice inverse trend to homing. So what this tells us is that when fishes are small and you put them somewhere else, they're more likely to stay there. But as they grow up very quickly, they become likely to come home. So as they grow up, this might be that they develop sensory abilities or inherent will to come home, essentially. But we see that shift in their behavior as they grow. And then we could look at the body sizes when this behavior of coming home becomes more likely. And we compared that to body sizes of settlement. So when these fishes settle onto the reef from the planktonic stage. And we found through comparing those body sizes that these fishes become sticky and attached to their home really soon after settlement. So within days of settling and growing a little bit, these fishes start to home. So in the bigger picture, what this tells us is that fishes really care where they are. They don't just randomly live somewhere on a reef that humans think is suitable to them. They have a particular home. And what this means is that we have these sticky fish communities, which potentially could be a problem for that connectivity I was talking about, because if fishes are so attached to a certain location, then maybe they might not be as likely to pick up and move to a better site. But also this homing and this displacement is unnatural. So I displace them and nature doesn't really do that. So I was also interested in what happens if fishes have to move or if they're forced to relocate. And that came up again through this natural experiment of the big bleaching event. So this is the images I showed before. This is on Lizard Island, exactly the same site throughout the bleaching event. And this is actually part of a data set where we have, I forget now, but a few hundreds of these locations. And then throughout our data, we can track the reduction in coral through the bleaching. And we can also track the abundance of the coral associated fishes. So this here is the coral cover. We have Acropora. So that's one of the main branching preferred habitats of these coral associated fishes. And we have the change in cover on the y-axis. So you can see, oh, and the different bars are just different quadrats, different locations, and they're just ordered by magnitude of change in cover. So you can see we had some significant losses in some of the sites. Up to 80% of coral, of Acropora coral, disappeared during the bleaching event. And because these fishes are obligate coral dwellers, and we think they really need that coral to survive, we would expect the same trend to happen with fishes. However, we found this. So we found this, what we call the spatial mismatch in coral loss and fish abundance. And what that means is that either this habitat dependence of these fishes on the coral might be lower than we think, or alternatively, these fishes might have a higher movement capacity than we think, because usually we see them associated with these corals, so we don't think they move much, but actually they might have the ability to move quite a bit. So this was a study we did based on these observations of these patterns on lizard during the bleaching event, but I wanted to understand a bit more about the behavior that sits behind this. So for that, I wanted to track obligate coral dwellers. So I did this on Pomocentris molucensis and Chromis viridis. And basically, I wanted to know whether they behave differently in good and bad neighborhoods. And what I mean by that is this. So we know usually we've got live coral, branching coral, that's where these fishes like to live. And that's where we usually see them in the vast majority of cases associated with these corals, sitting within the branches or just above. And so that's why we call them obligate coral dwellers, because that's where we see them. But then what happens if the coral dies? Do the fishes stay at the coral, or do they change their behavior? And do they maybe move further away? Or do they maybe behave a bit erratically because they're looking for shelter from predators and they can't find it, so they might move a bit more dynamically around? And since that shift in behavior is inherently spatial, so either they move further or they move in different patterns, that's why I wanted to track this with spatial tracking of their behavior. But how do you track fishes that are so small? Because these fish are, the body is smaller than those acoustic receivers that we saw earlier in the telemetry studies. So we had to find a different method of following these fish. And really by coincidence, I stumbled into this methodology. So that's 3D photogrammetry, where you can take images of the reef and then with computer software, you can stitch it together and you get this three-dimensional model. And the biggest effect is that it looks really cool in presentations and you can show really cool videos with this. But what you can also do is you get these really cool maps. So on the left here, we see a so-called auto mosaic, which is a geo-referenced rectangular map, basically like a satellite image of a reef area, whatever size you want. And you also get the 3D data behind it. So each pixel of your map, you get an elevation data set, so you can calculate slopes and structural complexity and so on from that. For example, to measure how much coral cover there is. So I found this by chance really at the start of my PhD, and that was really useful for this fish tracking project. So what we did is we went underwater and we drew these mud maps on underwater paper and then recorded each fish every 15 seconds and drew the fish location on a map. And that feels very low tech, but because we had those really good maps as well, we could overlay our underwater paper onto the actual digital maps. And you can see here that Chris, who was my volunteer, Chris Hemmingson, he did a really good job because it matches almost perfectly. And then say this is one individual that we tracked on that day, and we can then use this data from our underwater maps and transfer it onto our digital maps in the lab. And then with that, we can do really good modern spatial analysis without needing GPS or telemetry, because by transferring this on the map, we can have geo-referenced, so GPS coordinates essentially of that small scale behavior of these fishes. So what we did is calculate kernel utilization distributions, or essentially hotspot analysis of these usage areas. And we did that every day for 47 fishes across 6 days. So there was a lot of diving and a lot of dot drawing. But what we also looked at was in between days variability, because that would get to that erratic behavior that I mentioned earlier, that fishes might behave differently in low coral cover. So with that analysis, we wanted to see if fishes behave differently in high or low quality habitat. So these are some results. Here we've got Pomocentris molucensis and the core area, so the usage area on the Y, and 4 different sites on the X-axis. And the first 3 sites you can see, this is pretty much what we expect. So we have very small areas, less than half a square meter, where these fishes sit over 6 days. So they really don't move much. At this site 5, however, we saw this jump. So suddenly they moved further, almost doubled in their movement. And then this pattern was similar, if not more pronounced in Chromis viridis. So here I just want to point out the axis here. So this is 100 square meters. This is far, far further movement than we would have expected of these small fishes. And you see both of these jumps suddenly in movement were at this site 5. So something about site 5 must have been driving this. And we thought, it's probably habitat quality. But just to put into perspective how far they really moved, here's the images of some sites. So that's site 1, 3, and 5, just as an example. And this is site 5 in sort of geographic orientation. And that is about the home range size that we expected of these fishes. However, these fishes, especially that viridis that moved hundreds of meters, moved along this entire reef crest. So we had to then, after they went moving, we had to go back and map even more of the reef. And those diver icons, they're about approximate to scale to my body size. So you can imagine a fish that's this big moving all that way. It's quite a journey. Actually, that area they moved over is equivalent to parrotfish home ranges. And here's just the size comparison between parrotfish and chromis. So you can see those little fish really can go quite far. So as I mentioned, we wanted to know if that was habitat quality that was driving this different behavior. And we had the 3D data. So we looked at these measurements of habitat quality in terms of variability of surface angles. So it's just basically bumpiness of the reef. Rugosity ratio, which is a measure of, how much branching corals do you have. And we also got live coral cover data from the images that are on the right side of your screen. And that's what I'll present now, live coral cover data. So here we've got 6 panels, and each panel is for a different body size of a coral reef fish. And the y-axis is again, the used area of the fish, and the x-axis is live coral cover. And as we said earlier, we think these fishes are obligate coral dwellers, so they need live coral for shelter from predators. So we would assume that especially the smallest fish would have the strongest response. So we would think that either with more coral, they move much further because they're not constrained by having to go to this one shelter location, or alternatively, in high coral, they don't move as much because they don't have to dart around to find shelter. So either way, we didn't know what to expect, but we expected a really strong response of the small fish. But that wasn't true. This is what we found. So no response, especially in the small ones. In the biggest fish, there was a bit of a response, but that was not significant. And you also see the spread of the data, so it's not really a very tight fit. So this was really surprising. And this was not only true for coral cover, but also for structural complexity data. So what does that mean? Well, we think it means that these fishes might have higher spatial flexibility than we think, because their movement behavior and their behavior in terms of using space was less closely tied to coral cover, what we would assume based on their observations of them living in corals. And really, if we think a bit more what that means, I think that means that we have to reassess what habitat dependent means, because when we go out on the reef now and it's a stable reef condition and we always see these fishes with these corals associating, we think they depend on that coral. But we won't know if they really depend on it unless the coral disappears. And then when it disappears, they might only have a preference for it, but they might also be able to live in other coral. So I think that's again where it comes in, that this dynamic shifting of reef condition is really important and is really a new space for us to think about these established assumptions we have about fish. So we've looked at these 3 different aspects of space use for now. We've looked at home ranges, mobility through space, and mobility through time. And to bring it back to me again, so this is my personal mobility over long spatial scales and long temporal scales as well. This is a bit zoomed in, and you can see my hotspots in Townsville. And right now, I'm in the ATSIP building, and I'm in room 231. So you know exactly where I am, you exactly know my presence. But to be honest, that's not interesting. And that's sort of what we do with fish as well. We look at their presence a lot. But what should matter more maybe is their function. And in this case, for me, my function right now is to talk and tell you about this, but it's not about where I am. So if we think about function in fishes, what we do and what my lab does and sort of my upbringing in science, we think about herbivores. So we think about feeding in fishes. So, but how do we quantify function? How do we measure functions? So what we do on a regular basis is we go out, we go scuba diving, and we do transect and we count fishes. So in the image on the right here, you can see a bunch of parrotfishes, which are important herbivores. And you can go along and you count them all. And then you can tally them up and you can count how many individuals are in a certain transect. But you can also count how many species there are. And you can also count how many functional groups there are. And functional groups are essentially just super clusters of species based on what they do. So in our case, in herbivores, there's different functional groups based on what type of algae they remove and how they remove that algae. So that's the ecological purpose, if you want. And then with that data, we collect this very often. It's really good to get an idea of what's there. So we do these transects on reefs, we count fishes, and we know what type of algae they consume. And then we can make these tallies and we can count per reef how many fish of each functional group do we have. And in this made up example, you would see that reef 1, for example, you would consider that more stable or more ecologically resilient because you've got 3 different functions in quite high abundance. And reef 3, in this case, you'd be a little bit worried because it only has really 1 function left and not as abundant. So this is the kind of data we collect. And I think that's really important and very useful. But what we kind of do with this is we extrapolate functions over space. So we see the fish and then we assume, okay, it's here, so its function is also here. And it's never quite explicitly explained over what area we assume the function exists. But implicitly, we assume it's either the area of the transect, or if we have that data, it is the home range of that fish. So I think that's what we're doing. And I want to talk about a bit what I think our assumptions are behind that. So if we do that, if we count the fish and we know what they do, they feed on algae in different ways, and we know roughly where they are. And then we basically assume that the reef is healthy if we have many of them. What we assume behind that is that fish are feeding like pancakes. So we think that their functional impact is spread around the reef or even in their home range, and is just filling the reef like a pancake, like a nice circular functional impact. And because we're not only thinking about one function, but there's many different functional groups that we see on one transect, say, we kind of treat the reef like a pancake stack. So each pancake layer is a different function. And if we see those fishes all in one spot, we assume that all these different functions are delivered in this one location. And then we essentially get the most comprehensive removal of algae. And then the reef would be the most resilient or most healthy because we get all these different ways of keeping algae under control. And we don't just do that in one location, but we do it in different locations and across time as well. So in a way, we have this massive pancake stack of functions on reefs. But what I'm interested in is, are these pancakes really round and neatly stacked? So do fishes really feed evenly? And do these different fishes really feed on top of one another? And to find that out, you have to look at where fishes are, but you also really have to look at their behavior because you have to understand where they feed. And that is a bit harder than counting presence, because for behavior, you need to have a bit of longer observation underwater to see what they do. And it's hard because you don't really know where fish will move, so you can't really follow one fish around and watch it feed. You can, but that's time consuming. But what you can do, what we've done, is we can put cameras out on the reef. And we didn't just put one camera out, but we put many cameras out. And then we put our little coordinate markers. So you see those floating tags along the reef here. And then we also did this photogrammetry again. So we made this big map. And on this map, it's a whole reef site. So it's 6 meters by 6 meters. And it's got 8 cameras. And all these cameras look at each other. So the entire map area is being recorded on video. And then we can see when a fish swims through the video, we can triangulate its position relative to those coordinate markers that I've got in there, but also relative to the natural reef structure, because those maps also have that 3D aspect to it. So if there's a fish swimming through the video, say across this lump, if this lump is this lump, I can then triangulate its position visually on video and can put a dot on that map where this fish is. So that's what I spent about all of 2017 on doing, and manually tracked fish behavior through videos and maps. And these are some results. So this is the type of data you can get with this. So we have the entire herbivore community in that reef location over half an hour. That's what this data is. And it's subsampled every 3 seconds. So on the left, we see movement locations, and on the right, we see feeding locations of fishes. So that's really a behavioral map of that community. And what you see already here is that fishes tend to sit on the left side of these. The right is not really well used. And also you see differences here. So this is a movement hotspot and it's a feeding hotspot. But here, this is only a feeding hotspot, but they don't really move there. So this already tells us something about their behavior. So they seem to be darting in here and feeding a lot, but not really hanging out. So this is really giving us fine-grained behavioral information. But what I did with this data, I did spatial analysis again, and I did these kernel utilization distributions, so these hotspot maps. And this is the moving one and the feeding one, and the warmer the colors are, the more of a hotspot that location is. And with this data, you can really quantify the areas. And what you can see here already, this is far more concentrated than a random distribution would be. And then if we're really looking only at the red areas, so the hotspots, where most of the feeding and most of the ecological function is happening, we can also split it by functional group. So as I mentioned earlier, these are just different types of algae removal. And this is what this data is. So we've got browsers, brushers, croppers, scrapers, and suckers. And the names don't really matter, but it's just 5 different ways of removing algae off the reef. And you can see their core areas are really quite small, especially the scrapers down here. They only had their functional impact in these little areas. I then averaged that space of core feeding across all functional groups and all of my study sites. And I found that only 14% of available reef was used for that high impact core feeding. So only that important ecological function of algae removal was really compressed into only 14% of space. So that shows that the pancakes really are quite small. They only cover 14% of the plate. But what we also need to understand is, are they stacked or not? And the stacking is the overlap of those feeding areas. And we can quantify that too with this data. So if you imagine this is a potential overlap scenario, so the different colors are different functional groups, and it could look like this, just some random degree of overlap, or it could have these extreme cases. So we could have spatial complementarity, where fishes use spaces next to one another and don't overlap. Or we could have the alternative, which is essentially maximum overlap, where they all stack on top of one another. And that's the ideal pancake stack. And to quantify this, we can just count the proportion of area that is covered by 1, 2, 3, or 4 functional groups. So we can just count how much area is covered by 3 groups at the same location, what area is covered by 2 groups that overlap, and what area is covered by 1 group. And then we can plot that, and then with that we can do actual analysis. So spatial complementarity would have a pattern like this. We could have intermediate things, or we could have redundancy, where most of the area is covered by most functions. And this is the data behind this. So here is the same kind of graph. We've got feeding area on the Y and the number of overlapping functional groups on the X-axis. And this is what we found. So really, most of the overlap was only 2 groups at a maximum. So barely none of the reef was covered by 3, 4, or 5 groups overlapping over one another. But that alone was just the pattern we found. So we had to compare it to some sort of null hypotheses. And the first one was that fishes randomly feed in the available space. And that's sort of what we assume when we count a fish and assume, oh, it's on this transect, so it'll feed here. And if under that assumption, we would get this pattern. So we would just by chance see a lot more overlap in feeding areas. Another null hypothesis was this, and that's that fishes randomly feed in the space that they move in, that they occupy. And that is basically the assumptions of our fishes feed in their home ranges. But then we would see this pattern. So less overlap, but still more than what we actually observed. So this means that fishes seem to be acting in this spatial complementarity. They tend to be feeding next to one another, apparently. And if we zoom out, what that could mean is that we end up with these functional ghettos on the reef, where certain functions are only delivered in certain areas and is not evenly covered. And that could be problematic in terms of algal removal if some areas only receive that type of removal and others another type. So we don't have a pancake stack, unfortunately. The pancakes are more flung about the table. But really, again, zooming out a bit more, what this means is that fish presence does not mean functional impact. Just because there is a fish there doesn't mean it's going to deliver its function in that space. It's probably going to deliver its function in 14% of that space, but that's a completely different degree of functional impact. And then lastly, what we also wanted to look at is trying to identify what might be driving this pattern of low overlap. And for that, we had another null hypothesis, and that was that the area sizes within which they feed are stable. So the size of the area is stable, but the location is random. And that would mean that some external factors define the size of the area, but the location is actually stochastic, and that could just happen however, randomly. And with that null hypothesis, we found this pattern. So that was indistinguishable from what we found. So what we think might be behind this is that external factors compress feeding into these small 14% areas, and then the location is just random, but the compression is the problem. And we have first indications that things like sediment, for example, sediment and turf algae might be compressing that functional impact of fishes into smaller and smaller spaces. So what have we learned about fishes in space? So I think it's really interesting because it defines where fishes grow, eat, and survive. And if we care about fish on reefs, we should think about where they do all these things. So we've learned that home ranges exist and fishes have small and stable areas where they live. We've also learned that fishes can move and cover certain distances and have that potential of connecting habitats of different quality. We've also learned in sort of temporal connectivity that fish communities can be really sticky and not really move much, but they have their home and they stick to it. But then we've also seen that fishes that we think are really dependent on certain habitat qualities, that they actually might be a bit more flexible than we think so far. So there's a bit of hope there. And then finally, we've seen that functions and space is also a really interesting avenue to explore because we need to understand where functional impact is actually delivered. So again, space use in fishes is more than just measuring the where and measuring the home ranges. The presence of a fish is key because if a fish isn't there, it's not going to do anything. But once it's there, we also really have to look at its behavior because behavior really matters. For all these things, we need specific methods for all these specific questions. And that's what I mentioned earlier. Sometimes if you find a new method, like in my case, the 3D photogrammetry, that opened completely new ways for me to look at these things and look at different aspects of space use. So I think we should also just explore methods and then see what we can answer with them sometimes. And lastly, sort of the big picture thing that I really keep banging on about is that we really need to consider heterogeneity or patchiness of reefs and also connectivity of populations. I think that's the two big keystones that will help us to understand how reefs continue to exist in the future. And yeah, this is what I did in my PhD, and I'm hoping to do a bit more on some of these aspects. Like I'd like to do a bit more on that video and the functional feeding maps. But what I'm doing right now is far more theoretical, but still related. So right now I work on functional traits in coral reef fishes. And that's a very good buzzword. And I think many of us have an idea what it means. But essentially it's we can calculate what fish populations, what sort of traits they have. And if they're functional traits, we can then link from fish populations to ecosystem stability. So we basically want to predict if we have fish A, B, and C, and they have functional traits A, B, and C, then we can calculate if the ecosystem is going to be stable or not. But what I'm trying to do is understand why we really use functional traits, or really what's our motivation behind it, whether they really make sense to get to ecosystem stability, and how we can make functional traits a bit better and a bit more useful. But that's ongoing work, and I hope I can give a talk on that at some point in the next year, maybe. We'll see how quickly I can get this out of my brain and onto paper. All right, and with that, thanks very much. I hope you got some insights into space use, and yeah, feel free to ask questions now or email me or get in touch. Thanks.